Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Welcome everybody, I'm Pete Meyer from SB Grid, and so just some procedural stuff before we get started. The next webinar in the SB Grid webinar series is going to be December 10th at the same time, and that's going to be Ariana Peck, and she'll be talking about ARITOMO 3, real-time processing pipeline for generating 1,000 accurate and contrast-enhanced tomographs. For the session today, if you have questions, please feel free to use the Q&A button or send them to the hosts in the chat, and we'll moderate those. For today, we have Pavel Afonine, who's, if you're a crystallographer who's on any of the mailing lists, he probably doesn't need any introduction, but he's a computational research scientist at LBL, and today he's going to be talking about AquaRef, machine learning, accelerated quantum refinement of protein structures. So with that, Pavel, thank you for joining us, and I will turn it over to you.
All right. Thanks, Pete, for the introduction, and thank you, SB Grid, for providing me a forum to talk about exciting stuff that we've been working on over the past decade. Just making sure, can you see my slides? Can you hear me? If everything is all right?
Yep, slides look good. Audio seems okay.
Excellent. Thank you very much, all the audience, for joining this morning as well. Well, morning for me, or whatever time is for you. And yes, today I'm going to talk to you about AquaRef, which is AI-enabled quantum refinement of atomic models against crystallographic or cryo-EM data. And you see many people on this slide, so this is not just one person effort, it's a collaboration across multiple teams, which I will talk in greater details later on. And also, I'm going to really give you a very superficial overview of this. So I'll take baby steps to introduce you into this rather than go into great details, for which we would need to have way more time than 45 minutes that I have. Okay, so as I said, let's do baby steps and just get introduced and see what it is all about. Structure refinement. Structure refinement is, if you look at this pretty standard textbook picture, which shows you the pipeline of solving a structure using crystallography. And by the way, using cryo-EM is very, very similar to this. So atomic model refinement is just one step in this pipeline. And this pipeline in reality isn't as linear as I'm showing here, but nevertheless, model refinement is just one step in this pipeline that is next to the final step. And it is sort of critical because without this step, if you eliminate this step, you will be able to publish your structure. Because why? Because refinement makes your atomic model comply with community standard validation criteria that are necessary to pass another that you publish your atomic model and release it to the world. So all I'm trying to say here is refinement is totally critical step in this pipeline. Right, now a little bit more detailed view is what is refinement? Refinement is basically an optimization process of fitting your atomic model to experimental data. So it requires model, experimental data, some extra information to help refinement. And then the procedure iterates by optimizing a score that relates model to the data by changing model parameters with the hope that you get an improved model in the end, right? And just a pictorial illustration of that is basically map is your data, this is the model, and basically starting somewhere, model moves to the data, okay? So that's what refinement is about. And particularly in this pipeline, we'll be most interested in focusing on the refinement target function and restraints, right? So I mentioned on previous slide that refinement is all about optimizing some score target function, score function. There are many different names for the same, which is basically a mathematical expression typically containing two terms. The first term describes how well atomic model fits the experimental data, and the second term is necessary to compensate for the lack of infinite quality of your data. So your data is never perfect. It's finite resolution, contains errors, and so on and so on. So on data, the data are never perfect. And to compensate for that fact in refinement, we need to bring extra information. That extra information comes with a certain dose, with a certain weight. And that information is all we know about chemistry of your atomic structure, right? That restraint term parameterizes your structure in terms of covalent bonds and angles, dihedral or torsion angles, planes, repulsion terms, chirality, and so on and so on. And again, just to reiterate what I said, these restraints are needed because the data resolution is never perfect, right? So let's just have this again, textbook kind of illustration for those who are not very familiar with this topic. So one angstrom resolution, if you have a data at hand that is one angstrom resolution, you see individual atoms as peaks in your map. So you know exactly where to place those atoms in order to build an atomic model, right? Now, if you have anything like 2 angstrom resolution data, which is typical resolution in protein data bank, then you know where to put your model trace, but the individual atoms, or in 3 angstrom resolution map, the individual atoms are not resolved anymore. So those atoms can be anywhere in this envelope of density, right? And if you place them anywhere, then your atomic model won't make chemical sense. And again, and if you go even to lower resolution, kind of have this boomerang-shaped blob density where, well, you know what this model is, but you have no idea what the residue is, what its planar tyrosine, and so on. So the restraints are absolutely necessary to compensate, to maintain the sanity of your model. And again, just to convince you even more, this is just an illustration of refining with and without restraints. If we don't use restraints, and if we have the situation when we want to refine this model into this map, if we just turn off this, eliminate this term, this is what's going to happen. The atoms are going to rush into the density. They will fit the map almost perfectly, but the atomic model is not going to make any chemical sense, right? It's pretty obvious on this picture. While if we do use restraints and refine this model against this blob of density, during the entire process, the molecule does make chemical sense, as we can see here. Okay, good. Now, so I mentioned all these restraints on covalent geometry, which we refer to as standard restraints, which are absolutely necessary and great at typical resolutions which are about between 2-ish and 4 or 5. But as we go towards lower resolution spectrum of data resolutions, those restraints may not be sufficient, because if you look at this example here, we know what a molecule is, but we have no idea that, for example, this piece of molecule is actually alpha helix, right? So nothing in the map tells you this is an alpha helix. And so if we don't inform our procedures about this kind of information, this is going to be problematic. And again, this is another illustration of what I just said and why the restraint is important, right? So in this particular example, I taken perfect alpha helix, a perfect sausage of density, 6 angstrom resolution, and I try to refine this perfect alpha helix against this data without telling refinement this is an alpha helix, right? So the ions are going to move around, but as you can see, the alpha helix geometry isn't preserved during this process because we haven't told refinement that this is an alpha helix. All refinement knows, it knows about the standard restraints, bonds, angles, planes, and so on, and the map, right? So that's an illustration why refinement using standard restraints is problematic at low resolution. Okay, so however, we've been able to refine all the structures cryo, past cryo revolution against low resolution data. By low resolution, I mean anything lower than 3 angstrom. And you may wonder, well, if it's so hard to refine against low resolution, how we've been able to do that so far? Well, we use all this extra information about structures, right? So if there's internal symmetry in crystallography world called non-crystallographic symmetry, or NCS, we do use that. If there is a similar structure in the protein data bank, similar high resolution structure to the structure that you are actually refining, we can use that information as a reference for our restraints. We use secondary structure information, we use Raman channel plot information, side chain distributions as rotamers, and so on. So by using all this information, we can actually bootstrap refinement, low resolution refinement, and make it meaningful with a limitation that I will mention. And here we go. So all this extra information that I've just mentioned in previous slides are actually standard validation tools, right? So the Ramachandran plot, every time we talk about validation, the first model, atomic model validation, to be precise, the first thing that comes to mind is Ramachandran plot, right? Or secondary structure, or rotamers, side chain rotamers. Now, if we use this information as active restraints targets, those entities are not independent validators anymore because they're actively used in fitting, right? So we can still use them to validate our structures, but they're not independent and not as powerful as if they used to be before using them as actual active refinement targets. So this is the first problem. The second problem with this is that setting all these restraints is problematic and tricky because it requires semi-manual annotation. What I mean by that is how you tell secondary structure of your how you annotate secondary structure of your molecule, right? There are tools to just give it a PDB file and get a secondary structure annotations, but those tools rely on the geometry of your atomic model. And the geometry isn't perfect, the annotation is going to be not perfect. So garbage in, garbage out. And then it's on you to likely subjectively annotate your structure. And if you make an error in this annotation, that error will be propagated during refinement onto your structure, and that can be problematic as well. Now, and here's an illustration of this problem as a whole that happens in our field these days. So basically what we're looking at is the PNAS Structures 2019, right? It's published there, right? So high-profile journal, supposedly very good structure, and indeed the table 1 statistics is perfect, right? So it's really perfect. The low clash score, no Ramanujan outliers almost, and a few numbers fine. No random outliers. Everything looks perfect for the 3-angstrom resolution, except that if you look at the Ramachandran plot looks just wrong. Okay, and why is looking wrong is the subject of a different conversation on validation, so I'm not going to go into great details. All I'm saying is this is just plain wrong. And again, if you go back and look at that table 1 kind of extract, that looks picture perfect. So now to the very topic of my talk today is, I believe I just I made enough of introduction now. And so we go back to restraints, because restraints are responsible for all these gains and troubles that I've so far mentioned. And the standard restraints that everybody uses these days in refinement tools, not necessarily Phoenix, CCP4, Shellac, so all these, major players in structure refinement domain, they can be very, very same set of restraints that I took freedom to call standard restraints. And these restraints, I'm saying they are too limited. So why they're too limited? Here are the reasons. So there's no attraction term in this formulation, right? So we have bonds, angles, torsions, planes, chirality. We have repulsion. So two atoms, if they run onto each other, they will repel, they will pushed apart. But there's no electrostatics and there's no attraction terms. Here we're not using any information about protein structure, and this is limited to standard entities such as tabulated known ligands in monomer library or whatever GeoStandard and Phoenix libraries. And if you happen to have a novel ligand or existing entity that is chemically modified, you're out of luck, and you are on your own to provide that information to refinement, because otherwise refinement will see that new thing in your structure and stop saying, well, I don't know what it is. I don't know how to handle that. So again, these restraints are very limited. Now, what I'm proposing to do, we are proposing to do, the team of developers of this new method is to say, well, let's use QM calculations instead of standard restraints. So let's forget about libraries, all these monomer libraries, all these Hangen, Huber, tabulated bond lengths and angles, and instead let's use fundamental laws of physics to derive chemical information about your structure in terms of energies and gradients that can be used in restraints. So let's use fundamental QM calculations, quantum mechanical calculations, to replace the restraints that everybody has been using so far for many decades. So that's the idea. And you just may scream out loud saying, hey, Pavel, are you crazy? What are you suggesting? We all know that QM calculations are very computationally heavy, right? So they may take forever for even small, tiny molecule, organic or inorganic molecule. But here you're proposing, doing this for the entire protein, right? That entire protein could be ribosome, could be anything, and it can have lots of symmetry, right? So what are we talking about? Well, let's have a closer look at this and have a look at history of progress in the domain of QM calculations for large structures. So up until 2010, QM calculations for proteins were impossible in principle and were limited to very small molecules. So that's the knowledge we have, and that's all right and true. And that changed when the first revolutionary breakthrough happened when someone from Todd Martinez's lab in Stanford, the guy, his name Ivan Ofimtsov and colleagues, they coded QM calculations using GPUs in a very, very efficient way. And that was framed as a TerraChem software package, which is commercial, I think, at this point, so not even free for academics. And that actually made possible to do QM calculations for protein-like molecules. Yet again, these are protein-like molecules, very small kind of protein structures. So that was cool, and is cool, but still limited to those small molecules and probably, one or two single point calculations. And in refinement, we need hundreds and thousands of these calculations. All right, so another breakthrough happened in 2017 when members in my team actually managed to come up with a method to fragment a structure in chemically intuitive and meaningful way so that we can perform QM calculations for each fragment independently and then assemble back results into the whole structure. So that enabled QM calculations and actual refinement for real size protein structures. Yet that would require a lot of CPUs, probably a good computer cluster, and refinement times would range between many hours to days and even weeks for very large structures. And this was done actually, I will mention, this is where I'm going to mentioning who is involved in this. So this was done as a result of very interesting collaboration between crystallography software developers and methods, which kind of I'm representing this niche, and people having quantum chemistry, computational chemistry, general chemistry expertise, such as Mark, Waller, Holger, Malga, Nigel. Nigel actually also kind of does all of this. So we started this collaboration back in 2014 in a very interesting place in China, in Shanghai, back then, because we were offered space and funding to do this there for pretty much free from us. So back in 2014, we started that collaboration, and that collaboration really brought these very diverse people, crystallographers, software developers, chemists together. And that's a perfect blend of expertise to perform this kind of a project, right? And we know everything about crystallography, but for example, Mark doesn't even know what the crystallographic R factor is. But we know how to talk. We talk in English. And that really was productive in a way that we could blend our expertise in a synergetic way. So as a result, when we started that back in 2014, and back then when we started, we just made a list of things that we need to achieve in order to enable full model refinement using QM. So in 2016, we published a foundation paper that basically just describes our goals and means and targets. And then we started solving issues that are on the way one by one. So the scalability, as again, QM scales with the number of atoms very poorly. So we had to solve that issue. And we solved it by fragmenting models into small chunks in a very efficient way. That was published in 2017. And crystallographic symmetry, as you, I'm sure, all know, QM software is agnostic to crystallographic symmetry. So we had to solve that problem, and we solved that back in 2019 in a fundamental way that is encoded in CCTBX, Phoenix, and Quantum Refinement Project, and that was published again in 2019. Then, of course, we were not focusing on crystallography because crime revolution wasn't going around the time. So we enabled all of this for real space refinement, for cryo-EM, and so on and so on. So this is kind of a way, systematic, robust way that we took to achieve our goals. And in the end of the day, we realized that it all works, but it's still limited. As I said, you could refine the entire structure using these tools, but it would take a long time. And this is when we realize we need extra expertise. And that time of 2020, 2019 was the time when AI, field of structural biology kind of entered and kind of became the thing. So we realized, well, we have a very good expertise, us crystallographers and cryochemists and, theoretical chemists, but we also need AI expertise in chemistry. And this is when we discovered the team of Alexander Isaev and Adrian Royberg from, these guys are from CMU, Carnegie Mellon University, and Adrian is from University of Florida. So these guys are expert in theoretical chemistry plus AI machine learning. So we added them as an extra expertise to our bucket. Now we have crystallography, software development, QM, and AI, and that's what made the next breakthrough, which I'm talking about, is now with this expertise, we came up with a way to do QM calculations on your laptop for any size protein structure in a matter of seconds or fraction of seconds. And that's what I call revolutionary, because again, we went from 2010 impossible in principle to possible on your laptop. That's big, I believe. So, and that was thanks to kind of this sort of project management and bringing right people for the job together. Now, you may wonder how it all works. And as I said, with the time I have, I don't really have much opportunity to tell you a lot of details, but I will hint you how it all works. So, and the procedure is quite standard in AI machine learning approaches. So basically what we did, we took all amino acids residues, right? So 20 plus residues with all possible protonation states. As you know, some of the residues have different protonation states, such as histidines, for example, right? And we combine them as single residues, dipeptides, 3 peptides, 4 peptides. We included disulfides and so on and so on. So that's a lot of combination right here in this first step. Now, we perturbed all the structures in torsion angle space to generate conformational diversity. And this too means we have lot of conformations, lot of structures. It's millions and millions of structures. And now for each one, we will do DFT, density functional theory calculations, just single point calculation for each of these our million of structures. And remember the conformation and the energy gradients that is associated with that conformation. Now, what do I mean by remembering that conformation? And that's the key here. So we would have to parameterize protein molecule topology and chemistry in a way that you can fit into machine that machine can understand, right? And that's the general thing we do in machine learning. We need to describe our problem at hand into some way that machine understands. And the way we're taking here is described in this kind of inaugural paper, 2017, as basically, if in one sentence, basically this is an approach that lets you go from a molecule to a vector of parameters that describes topology and chemistry of this molecule as again a linear vector of numbers. And these numbers are very well understood by the machine, and these numbers can be associated with the energy gradients calculated at this stage. And tracked, remembered, repeated for next molecule, and so on. And in the end, and by the way, these calculations typically take about a week on one of the biggest national computing resource. So it's very, very, very hefty, very heavy calculations. But in the end, you get a black box. You get a machine learning model, which now you can take and given a model, a molecule at hand, you throw that molecule at hand into that black box. And almost instantaneously, you get values of energy and gradients, which correspond to QM calculations, except that these calculations take a fraction of a second. So that's the big picture idea. Again, I could spend an hour talking about it, but that's how much time we have for this. And so here I'm showing you it's an example of how long these calculations take. So this is time as a function of number of atoms in the molecule you try, and this is how much GPU memory it takes. So basically, if you just look at this, it's pretty linear, not quite, but what we really see striking here is that for something of the size of 100,000 atoms, it's a very extra large molecule, 100,000 atoms. The calculation takes about 0.6 seconds. So it's about half a second for something as large as this. So, what does it mean? That means we can actually run QM-based refinement on our laptop because this is the amount of time approximately that takes to calculate our simplistic standard restraints. Except that these restraints, these QM restraints, know all about fundamental physics. They know interactions. They know all about chemistry of your structure, and they do not require parameterizing your molecule. They do not require you telling the procedure, this is my ligand, these are bonds and angles, and this is how they're related. All is tedious and error-prone gel. So all is handled sort of naturally by the nature of QM calculations. And of course, right, so that sounds exciting, I believe, and now we need to sell this to the community. Not sell, right? We need to prove it works and publish. And as part of this, and I'm not showing, we did a lot of testing, and I'm just showing you a few tests as just a convincing point. So we tested at high and low resolution using cryo-EM and X-ray data. Here I'm showing benchmarking and testing against low resolution 3 angstrom or worse resolution of your data. Cryo-EM models, and actually I missed here. So we have 40 cryo-EM models in this test, and we have 20 X-ray models. So the 20 X-ray models is actually missing on the slide. So we have about 60 models total, and we selected all these models in a way that each of them has a high resolution homolog available in Protein Data Bank with the 95 and better sequence identity and greater than and better than 1 angstrom superposition OSD. And why we have this is because we're kind of assuming that those models are close enough to be considered as ground truths to compare against, because they are high resolution and they are very, very similar. And we performed the comparisons in torsion and Cartesian angle space. So now here's the result of this testing for this particular test. Again, we did way more tests. This is just one I'm showing you here. So basically, I will take time to explain. So blue is standard refinement in Phoenix. It's just a refinement you run normally when you define your structure against X-ray and cryo-EM data. And orange, or whatever the color it is this AquaRef refinement, the new refinement feature with QM-based restraints. So blue Phoenix standard refinement, orange AquaRef refinement. And now we're looking at the validation metrics you normally report in your table 1 structure. Not all of them, but just a few. So mole probability score. Mole probability score is basically accumulates clash score, Ramachandran plot, and Rotemer metrics into one metric, and it's kind of is related to the resolution. It's calibrated the way that kind of related to the resolution of your data. So the lower the number, the better. And as you can see, AquaRef refinements are substantially, very obviously better in terms of mole probability score. All the 60 refinements, 40 cryo-EM and 20 X-ray refinements, low resolution, 3 and lower, are substantially better. What that means, it means lower clash score, better fit Ramachandran plot, better Rotemer's distribution in refined structure. RamaZ is also very, very good. You get substantial improvement. And RamaZ is a metric that describes the quality of Ramachandran plot. For those who are not familiar, just Google RamaZ and you'll discover a very, very interesting topic if you're not familiar. And then all others, Kablam outliers and favorite and so on. And in terms of the fit to the data, because in this particular example, we are considering low resolution refinements, and those kind of refinements in using QM restraints, they're not expected to make a lot of large scale changes to your model. They're not supposed to change your R factors or cross correlation coefficients between maps substantially different. So basically all we see here is that those refinements reduce overfitting, right? In cryo, we don't have any R-free factors. We do not control overfitting in cryo. So this plot, for example, shows that the models in cryo field are typically overfitted against the data, at the expense of having poorer geometry. While if you use CAM-based methods for refinement, you get a better geometry at a slightly tighter fit to the data, which means, again, we improve geometry dramatically at the expense of a slightly worsening of this correlation coefficient, which translates into a clear statement that that means we reduce overfitting. And it's pretty similar for X-ray as well. Now, if you look at skew kurtosis plots of hydrogen bond parameter distributions, and again, for those who are not familiar with this, Google that. There's a publication and it's a standard method in Phoenix to evaluate quality of hydrogen bond parameters, which is basically just tells how your hydrogen bond parameters in the entire structure, how realistic they are. And very much like Ramachandran plot, there are counter plots for bond and angle of a hydrogen bond. And these represent probability distribution for this bonds and angles. So if your structure is inside the contouring, you're very good, probable. If outside, that's not very good. The standard refinement in Phoenix and other packages I'm not showing here, just to not criticize others, it's the hydrogen bonds are pretty rubbish and this metric, and using CAM-based approaches, they are quite well fit the expected distribution of these hydrogen bond parameters, derived again based on high resolution data. And this is a little bit busy plot, but it basically shows, remember in the beginning I mentioned all these 60 structures we decided to use as testing structures. They have high resolution homologous structures. And this is scatter plot showing the distance between refined structure and homologous structure using Aquaref refinement and Phoenix standard refinement in Cartesian space in angstroms and in torsion angle space in and torsion angles. And pretty much in majority, if not all, except for a few probably outliers, the refined structures with Aquaref got closer to the ground truth structures, high resolution homologous structures in both Cartesian and torsion angle spaces. So that's what this plot tells you. Now, and since this is actually the ratio of time taken by Aquaref or versus Phoenix. So time to run refinement using Aquaref divided by time taken by refining the structure using standard Phoenix refinement for all 60 structures, right? So this is this time ratio versus number of atoms in the 60 structures we used. So the plot is kind of telling you that the time is comparable one-to-one when the number of atoms below 10,000, and it can be 2 to 3 times slower. This is still something I cannot explain at the moment because I would expect the time would be kind of one-to-one. So I don't know why it's 2 to 3 times slower when the number of atoms grows. So this is kind of topic for ongoing research. So we are investigating this. So, but I'm just telling you the truth we have at the moment. So that's the matter of fact. But this can change over time as we investigate. Now, in terms of availability, Aquaref refinement is available as standalone. I actually don't have it on the slide, but you can install it using all the necessary information from Quantum Refinement web page, which requires a few steps, and that's best done by experts. and those people who like messing with software. So that's why I'm not advertising this here, but it's also available as part of Phoenix in a very streamlined way. So you just download and click and it's available. And so far it's available as a separate installers on Phoenix web page and that these installers are actually for Linux. It has requirements for CUDA 12 or 11. But if you fulfill those requirements and have that set up, you can download the software right now. As of today, you can just go and download it and install, and that will enable you to run these calculations on your own. So this limitation on availability will be lifted within the next few months as Billy Poon and our team making it more general and more streamlined. But so far, this is the route to get this working for you. Now, as a summary towards the end, and this is probably one of the last slides, as I also realize I'm at the time. So summary is CAM potentials perform amazingly well, and BNML accelerated by ML AI that makes it possible to run on your laptops. Current limitations, and this is probably the most important part. So this is protein only so far. So adding ligands and metals, that's work in progress. The protein only doesn't mean your input PDB file must have only proteins. It's just the calculations will be performed only for proteins, and your ligands will be kind of frozen in these calculations. Right, so no alternative conformations. Again, this is kind of hefty limitation for high-resolution X-ray structures because we always have alternative conformations. Important one, that's these calculations are intolerant to nonsense, which means what I really mean here is you shouldn't come in with very poor models that have very bad, severe geometry violations because QM are actually sensitive to this. And I think timing and hardware is something I mentioned just a minute ago, as well as availability. It's available in Phoenix right now in nightly builds. So if you want to try it, just get it from the nightly build, as this is work in progress. As I said, we'll be including different atom types and molecule types. So we'll leave the limitations of being protein only. But to be on bleeding edge, use the nightly builds of Phoenix to access the software. Just an acknowledgement sides to all the collaborators at Phoenix. I'm from Phoenix team, and several people were involved in Phoenix as well as outside Phoenix that I mentioned previously. And again, I thank SB Grid for providing this forum to inform you about this very, I believe, interesting developments. With this, I finish, and I thank you for your attention, and I'll be happy to answer any questions you might have.
Thanks, Pavel. That was really cool stuff, and we've had a couple questions come in from the audience. As usual, I have questions ranging from philosophy to implementation details. But I guess so. One of them that kind of stretches both of those. So you mentioned needs a GPU. It can run on CPU, but it's slower. It is with some of the ML toolkits that have been getting more wide use. Is there any prospect for different accelerators like Apple's M series or AMD, or is this planned to be CUDA only, or you can use a GPU or you can use a CPU, but if you're willing to wait a very long time?
Right. So technically, we just decided to be very careful and released what's on Phoenix webpage is bundled and released for Linux, CUDA 11 and 12, just to be safe. But generally, the machinery we use it does not limit you to use it on different flavors of that or using CPUs. So if you choose to wait longer, or if you have a rather small molecule to refine, and which might be okay to do on CPUs, that should be possible on CPUs. It's just at the moment we bundled that on Phoenix End for this particular setup, but generally at Phoenix it will be available across all the other platforms that Phoenix is supported on, and you don't have to actually stick to Phoenix to do that. So the actual software is you can get it from qdify.com page and GitHub page. It's just there are instructions there. It will be just you are on your own to bundle the things together and install, assuming your current computational setup. And then you got Phoenix the usual way, and it all should play together. It's just this kind of route that I'm talking about is more for the computer geeks people who like messing with doing. But it's not rocket science. It's just the multiple steps of following and having all these dependencies. And so again, it's not rocket science. It's just probably maybe way too much for a regular biologist, structural solution person to do. So that's why we made it available just with using this narrow avenue in Phoenix at the moment. So that will be expanded to broader range of platforms and configurations. And if you're kind of able to do it on your own, you can always get all the pieces and have your setup right.
Cool. Thank you.
Welcome.
And so is it still necessary to tune the weighting between the ML parameters and the experimental parameters, or is that just kind of like magically taken up in the mix?
So the way it's set up, the weight is calculated automatically. And for all the 60 examples I showed on the slide, the automatic setup works. So if you take any of the 60 examples, you don't really need to change anything. You'll just give it model and data, and refinement will work. However, as you can imagine, this is very, very fresh and ongoing. So I can imagine there might be situations where the weights may not be optimal. And then there are parameters in the software that you can actually change the weight. So, and how the weight isn't optimal, this is standard kind of symptom check. You get too large difference between R-work and R-free, or geometry becomes distorted in a way. So, and based on that, you change the weight to either favor data, experimental data, or restraint. So the usual way in refinement. But generally, we are trying this in this here. We're trying to make weight choices automatic, because it's just too much to ask people to find the best weight, because that means you need to run multiple refinements and see which one works best, and that's just not something people would be up to doing these days.
Yeah, there's always the trade-off between, and there's a manual knob, that means there's a chance to get it wrong. But
absolutely, yes.
Yeah. When you were talking early about what I thought was the very cold method stuff, so should structural biologists worry about the difference between quantum mechanics calculations and density functional theory calculations that you're fitting the parameter, the ML thing to, or is that we can just treat that as the other side of the black box?
Well, that's a very good question. And before we arrived actually to doing this, there was multiple publications, mostly done by folks I mentioned at CMU and University of Florida, Edgen and Alexander. they actually did a lot of benchmarking, a lot of tests and calculations, and they published all of that, demonstrating that the accuracy of Anderson gradients and optimized structures coming from pure QM as it is versus the machine learning approach, this machine learning black box. And depending on the level of theory, the accuracy can be from moderate to very good. And what we use here is basically one-to-one correspondence. So you can treat results that you get out of this black box is pretty much as if you just run the DFT calculations in any of available software such as XTB, I guess Orca, TerraChem. So bottom line, in one line answer is you shouldn't worry about matching actual DFT calculations versus this machine learning approach.
Well, I guess maybe I asked the question wrong. Like, is DFT and quantum, are those functionally equivalent? Because I'd been under the impression there was some difference in a way that I didn't understand, which is why I'm asking what may be a dumb question.
Well, I'm not an expert to answer this question, I guess. I'm not a QM person generally, so I don't think I think I can answer this. I mean, I have my idea, but I want to spread nonsense just in case I'm wrong. And yeah, I don't have an answer for this. And
the way I would bet then would be I'm asking a question that's a colloquially phrased, that's a dumb question, and we shouldn't worry about it.
No, it's not a dumb question. It's just I don't have an answer for it because again, like I mentioned, this collaboration, the beauty of this collaboration is that we have very, very broad expertise, but we are not experts in each of these things, right?
Yeah.
So if Alexander or Adrian were here, they would answer right away, instantaneously, this question.
Yeah.
But I don't have, and the same goes to the other end. So if you ask them anything about crystallography, they won't tell you anything because that's not their thing. All in all, yeah, I'm sorry, I don't have an answer. I don't have a good answer to this.
That's how collaborations work, and that's kind of how they should work, right?
Exactly.
Awesome. All right. So let's see. We have another question that came in from the audience that I'm trying to read through here. So I guess paraphrasing slightly, is there actually? I guess perhaps I'll just read it rather than trying to paraphrase. So, is there a way that we could make the ML models handle ambiguity in the experimental data here during refinement? And are there ways to optimize the ML models for sparse experimental data?
Oh, okay. Well, I don't really. Well, I think I don't really fully understand the question. So, I mean, the ambiguity in experimental data always exists, and what is ambiguity in experimental data, right? So, are we talking about things like alternative conformations, right? So the data tells you this atom either here or there with certain probability. So this can be thought of as an ambiguity in experimental data, or talking about errors in experimental data, right? So the measurement errors, that's another kind of ambiguity in experimental data. So I don't think the question is clear about this nuance. So if you talk about experimental data errors, that is handled by things like maximum likelihood refinement targets, and this is totally separate thing from restraints and this QM stuff in general. So the maximum likelihood refinement targets, we still use them regardless the kind of restraints we use. If we talk about ambiguity in experimental data as alternative conformations, then as I said, in this approach, QM-based, QM refinement approach, we don't handle alternative conformations just yet. And this is one of our items in to-do list to address. And that we'll be able to handle that. My prediction is within next 3 to 4 months. So we'll handle that for sure, but not at the moment.
Well, thank you. And Muskan, if you'd like to clarify, please let me know, because I think that addressed your question. And yeah, so if I guess one final one is, this is more on the philosophical side, but it seems like both AI has been referred to as like a universal function approximator, and Fourier transforms have been referred to as universal function approximators. Like, is that just convergent evolution, or do you have any thoughts on that, or is that another incoherent question?
Well, it's indeed a philosophical question, and guess that requires a proper setup to answer it. Maybe a pop with a beer. Well, that's a true statement, right? So we can use AI to, if the problem is well defined, not ill-posed, then likely with enough data it can be mimicked, approximated by AI. So this restraint stuff is a perfect example of how that can be done.
Yeah.
Well, AlphaFold is another example. So I guess it's literally down to the point whether your problem at hand is well defined and has enough data. And if it's well defined and has enough data, it most likely can be approached by AI, and that's how it works. Again, AlphaFold and the Aquaref requirements are one or two examples that illustrates this. So yes, the answer is yes. It's kind of along that way.
Great. I guess we're running out of time, so I guess one last call for quick questions, and looks like no. So Pavel, thank you again. This was a very cool talk. Not so much a question, but this is impressive stuff.
Thank you very much. This is ongoing. I'm sure in a year from now we'll be able to do ligands and other stuff. So this is really pacing very, very actively and fast. So yeah, stay tuned, guys. Thank you very much.
And I mean, a factor of 2 for being slower for quantum mechanics equivalent refinements, like that is super fast. So.
I'm almost sure we'll get rid of that factor too.
yeah, but it's still amazingly fast. So cool.
Thank you.
Well, thanks everybody, and enjoy the rest of your day in whatever time zone you may be in.
All right. Bye.